Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Jeremiah Sulam, Assistant Professor of the Biomedical Engineering Department at Johns Hopkins. He is also the recipient of the 2022 Toffler Scholar Award. I'm looking forward to hearing about Dr. Sulam's effort in developing more accurate and efficient imaging of the brain. Let's dive in. So welcome.
Thank you. Thank you for having me.
I've been looking forward to this discussion all week. This is what you're doing is absolutely fascinating, and looking forward to this discussion. So, first I want to kind of explore maybe, kind of take me back and set the foundation here of your formal education. You have a degree in biomedical engineering and you hold a PhD in computer science. Tell me, what is biomedical engineering and how does that relate to computer science?
Terrific. That's a great question. So I think that biomedical engineering is really about understanding problems in medicine and health and looking at them from an engineering perspective. So trying to identify problems and trying to come up with solutions to those problems. I was raised in a household with a mother who is an MD. My mom was an anesthesiologist, recently retired, but my father was an engineer. And so, over dinners, we would often talk about some of the issues that my mom, would find in the operating room. And then my dad would comment on some of the problems that he would face at work. And I guess I sort of grew up with this kind of analytical way of thinking about the world, but also being very much interested in how our body works. And so I think that naturally delved into me going into biomedical engineering as an undergrad. I remember sort of flirting with the idea of going into medicine, but my mom wasn't really a fan of that idea. And for whatever reason, she would much rather prefer I became an engineer. So I did biomedical engineering as an undergrad. This was, by the way, in Argentina, where I'm originally from. And sort of towards the end of my degree, I was getting increasingly interested in providing solutions to medical problems that relied on using computers and to using automatic procedures for answers. So the way we do this is we try to find problems that have some patterns that we can leverage. So oftentimes there are problems in healthcare whose solutions relies on us following specific rules. These can be, for example, the steps that a clinician follows when carrying out a diagnosis and then recommending a specific treatment. And in some cases, those procedures can be made automatic by means of computers and what we call algorithms, which is basically a pre-specified rules that computers follow. And so I was getting increasingly interested in this ability of us to provide solutions by using techniques in computer science. And so from then, it was more or less natural for me to try to further my studies and go to grad school in a computer science department. And this happened to be at the Technion in Israel, where I went for grad school. So I sort of had a lot of fun working in a computer science department, really, learning a lot more about the CS world. But then when I was finishing my PhD, I was still very much interested in biomedical problems and in biomedical data science. And so eventually ended up finding my way here to Johns Hopkins, which is kind of like this perfect playground for us to explore solutions to pressing problems in medicine, but with a very strong engineering background, with a very strong focus on technology.
So thank you for going through that. And as you were kind of describing that, I was just thinking about how that technology, both in understanding the body, but also the computer technology and the algorithm software technology. How did that change through that journey of getting your PhD? Because there's been rapid advancements in the last decade or so.
Oh, yes, absolutely. It has changed drastically. So I began my PhD studies in 2013. And at the time, I was working mostly in computer vision problems, which are basically sort of designing and understanding algorithms to do certain things with images in an automatic way. So, for example, make them look prettier, to restore them, perhaps to remove some noise that has corrupted this image, and to do a bunch of things that users often want to do and need to do with images in their computers. At the time, we were using a set of tools that are very elegant mathematically and that were really very, very useful in practice as well. But 2013, for those who are aware with the recent changes in artificial intelligence, 2012 was kind of like a big year for the computer vision community and for the machine learning community. It was a year that a deep network won the ImageNet competition. And so from that point on, the attention towards these other tools based on deep networks became ever more increasing and hard to ignore in some sense. So a couple of years into my PhD, I sort of moved slightly away from some of the more applied problems that I was working on and started focusing on these new tools, on these new deep learning tools to try to understand them better, still by using some of the core elements that I was exploiting in my PhD, but try to bring them to bear in that world of deep convolutional networks and try to say something about, for example, when they would work, when they would not work, when these algorithms would be stable to perturbations and the like. So it has definitely influenced the kind of problems that I've been working on for the past 10, 15 years.
And for some of our listeners that may not be familiar with deep networks or deep learning, can you go through that a little bit?
Of course, these terms, and thank you for the opportunity also to clarify these, because these terms are often tossed in mass media and they appear everywhere, and sometimes it's not completely clear what we mean by these things. So neural networks is a model to learn algorithms from data. So these are functions. These are a set of instructions that are executed when someone instructs them to, when someone presses a button, for example. And neural networks have a particular way of coming up with these functions that are executed. They are really nothing else than linear transformations of your data. So you can think of these as basically matrix multiplications. You're multiplying a matrix by a vector, adding some offset term, and then having a very, very simple function that is slightly nonlinear. So it doesn't behave linearly. It's not just like a linear function. And this is a very, very simple structure that then is composed in a hierarchical manner. So in the end, these neural networks are simply a composition of these functions that are very, very simple. They're just linear or affine functions composed with these entry-wise nonlinear activation functions. They are often called neural networks because back in the 1940s or so, this was basically the working model for how neural networks in the human brain worked. That has, this neuroscience community has learned lots since then. And so now, these models are, it's pretty much understood that this is far from the way that neural networks in biological settings actually work. But we still call them neural networks. And this deep learning sort of revolution is simply the realization that when you compose these functions over and over and over again, you end up with these very deep hierarchical structures that, given enough data and given enough compute power, computation, you can end up actually learning very complicated functions in ways that was quite honestly surprising for most of the community about 10 years ago.
So the way I just understood that is the power of the computer, the power of understanding how things are connected and how this data relates to each other is it will bring new learning that maybe we wouldn't have seen if we were just kind of going through the data ourselves. So the computer is our partner in helping kind of unravel that complexity.
That is true. I'm not sure if I would say unravel, but I would say at the very least leverage. So let me give you an example. Let's say that you wanted to detect objects in videos from street images. This is a problem that the computer vision community has been working on for decades. The traditional way of solving these problems was to come up with some heuristic notion of what to look for in images, right? So if you were looking for example, people on these videos, then you could come up with essentially templates of what a standard person might look like on these videos. And then the task would basically boil down to looking for things in these images that resemble the template that you have. And maybe you would do all kinds of things. Maybe, for example, you would look at this template across different resolutions or across different scales, because people in a video could appear closer, could appear further apart. And so you want to be able to detect all these things. And so you may look for this template across a number of scales. Or you could also jitter these templates to accommodate for people in different orientations on the street. Maybe you would consider different color attributes that might characterize people. So all of these techniques worked okay, so to speak. But what these deep learning architectures brought that was new was the ability to basically put on the back seat all these work, all this sort of human intuition that had to go into coming up with these features. And instead, it allowed us to find these features in an automatic way. We now don't need to specify what these things should look for in these images, and rather these patterns of what characterizes something, for example, the presence of a person, are found automatically from the data. So sometimes we might have a good intuition as to what these features might be, but the features that are found by these models might be close to that, might not, but might be further from that. But in practice, these features tend to work very, very, very well and much better than what we were able to do before, basically by a more standard engineering approach of just telling these machines what to look for.
Yeah, that does, and that kind of leads me to you and your team just recently published a paper titled, and hopefully I'll get this correct. Brain imaging techniques allows researchers to achieve more with less. Basically, you came up with this ability to create these like super scans of the brain. Can you talk about what this publication is? What was the science behind it? And just really kind of the breakthrough? Because it sounds really interesting and exciting.
Sure, I would love to. So, this project is part of a large consortium that I started to get involved with when I joined Hopkins in 2018. And it has to do with providing ways for clinicians and MRI experts to image things faster and in more tractable ways, more comfortable ways than would otherwise be possible. So in this particular setting, what we are doing is we're looking at MRI imaging, magnetic resonance imaging, and these many of the listeners will probably have been in one of these MRIs. These are basically machines that get to image the interior of our body. They image human tissue by measuring different properties that our tissue has. In the specific paper that you're talking about, the property that we were interested in is called magnetic susceptibility, which is basically a physical quantity, just like mass or density or any other property of matter. But here, this magnetic susceptibility really measures the extent to which any piece of matter is magnetized when it is put in an external magnetic field. And it turns out that this is a very useful property to image because different tissues have different values for this quantity. And so if you get to image this property, you get an image of different tissues in the brain. This is particularly important because one of the main factors that contributes to the magnetic susceptibility of the brain is the myelin sheets of axons. So the axons are basically these long segments of cells. These are long segments of neurons, which are these brain cells that are responsible for conducting electricity and for all our cognitive processes and sensory processes. And these axons, in order to conduct impulses and to conduct signals better, are often wrapped around a sheath of myelin, which is basically a high density lipid sheath that wraps around the axon. And this is fundamental to the healthy conduction of stimuli in axons. This myelin, because of these high compressed lipid sheets and layers, has a very particular influence in the magnetic susceptibility that we can measure. And so if one were able to measure this magnetic susceptibility through an MRI machine, then you would get some very useful information about, for example, how healthy these myelin sheets are, on top of many other structures that you might want to image and many other factors that you might want to study in the brain. So this technique has sort of been known for about a decade or more. But the problem with this is that because of the physical properties of tissue and because of the way magnetic resonance works, it turns out that in order to reconstruct these images of the brain, one needs to ask the subject who is in the scanner to actually tilt or rotate their head inside the scanner and repeat the scanning process a number of times with their head at different orientations. And you could think of this as a way of getting a different look at what is in the brain. So if you were only taking one scan at a single orientation, there would be things that you would miss. And so by asking the patient to rotate their head inside the scanner, you get more measurements about the same underlying phenomenon. And then the hope is that you can put together all those measurements into a very clear reconstruction of the image as a 3D volumetric image of the brain. And so in the paper that you mentioned, what we were trying to do is reduce the number of those measurements that are needed to produce these reconstructions. So this is not too far from those original problems that I mentioned I was working on in the beginning of my PhD, where we were trying to basically remove corruptions from images and perhaps complete pieces of images that were missing because we didn't get to observe. At heart, the same phenomenon is at play here. There is information that we don't get to observe directly. That is why we would ask the patient to rotate their head. But we're trying to do that automatically. We are trying to reconstruct these missing pieces in an automatic way by having learned from data on how those missing pieces look like. So this is basically merging ideas of variational problems and inverse problems, which is a relatively mature field, but also leveraging these very powerful machine learning methods like these deep convolutional networks that we were discussing before.
Yeah, so that's the connection, is the advancements in this deep learning and deep networks that are helping identify that data that might be missing.
Yes, and more than identify the data that it's missing, what we are really using these networks for is to understand what is an appropriate model for the data that we are trying to reconstruct. So, for example, let's consider the case of trying to inpaint a part of an image. So, for example, imagine that you went on vacation and you likely at some point took a picture of a friend of yours and someone stepped in the image. A person who was just walking by. And now you're stuck with this image with your loved one, but also with this other person that just happened to have walked into the picture. So it would be amazing if you could just crop this person out and then inpaint in its place the background of whatever was behind this person. So naturally, this is essentially impossible because you did not get to image that background part. However, it's not hard to imagine that you can come up with a pretty accurate guess of what the background was. because you have an image of the world, you have a model of what the world looks like, you have information around that person that will help you infer what is behind that person. And so even though you cannot you cannot solve that problem exactly, you can find feasible solutions to that problem by understanding what the world is or by having a model of what the world looks like. So this is not too different from what is happening here in MRI. Here we have measurements that are incomplete. We have parts that we don't observe directly. We perhaps observe indirectly, like in the case of the person, you would observe the background surrounding that person, and that will give you information about what's in it. So in the same manner, when we have a patient inside the scanner and we acquire one or two of these orientations, we have an incomplete, imperfect picture of this brain. And then by having learned a model of what brains look like for example, a healthy population, then we can fill in these gaps and provide a better reconstruction that uses not only the measurements that we've obtained, but also everything that we've learned about what brains look like in a population of patients.
So why is that important? And especially given us trying to understand the brain and some of these horrific diseases of the brain. Why is this important?
So this is really fundamental to try to develop better biomarkers. So you're talking about diseases, for example, and often for those of us who work in technology, what we care about is understanding features or aspects or quantities that we can measure from data that can tell us something about, for example, the progression of a disease or the risk of a disease. And this is what we call the biomarker. It's just simply a number. It's something that we can measure that tells us something about the risk of a patient or an estimate of disease progression in a patient. So this modality of measuring magnetic susceptibility in the brain, because of all these nice properties that I was describing and its interplay with imaging myelin, could develop, and it's been sort of well understood that if we had access to a technology like this, we could develop a lot of very interesting biomarkers that we could use, for example, to track and estimate the status of health of the myelin sheaths, for example. But because this technology hasn't really been deployed to a state where it is comfortable for patients in the clinic, this has basically prevented the development of these potentially great and insightful biomarkers. And so the reason we think this is important is because by speeding up these scans, by requiring only a subset of the orientations that you would have had to acquire in the past, we can now go on and collect more data. We can start carrying out larger studies potentially, in the hope that we can find these biomarkers that allow us to understand the health status of the brain better. Or when you have diseases, in particular diseases that affect the myelin sheaths severely, such as multiple sclerosis, hopefully we can learn something more about these diseases through these new imaging techniques.
So what are the challenges in what you're doing and the research that you're doing? Is it the technology? Is it is it thinking through how this technology would go to market, across those institutions that have MRI machines? What are the challenges here?
So there is a significant number of people that are very excited about this. I work with colleagues at the KKI Center in Johns Hopkins. This is the Kennedy Krieger Institute. And in fact, I only got to learn and get into this problem because of my collaborators, because of Dr. Shu Lee, Dr. Peter Van Zykel, and many others who have been fantastic. So the main challenges now is in carrying out larger studies to really understand, to explore potential biomarkers that we can find based on these new imaging platforms. But at the same time, there's an important work that has to do in validating these techniques. So the validation that we have of these methods that have gone in the paper show pretty robust results, and we are very happy about them. But they are over relatively small number of patients. Because at the moment, remember when I was describing this acquisition process where you have to have patients inside the scanner with their heads in different orientations, this is a very time consuming, it's a very expensive process. And so the number of studies that we had access to is relatively small. So we are looking at expanding these validation studies, potentially including, for example, validation in animal models. There are different kinds of techniques that one can use to try to validate these techniques. But in a nutshell, that is the main challenge that we are facing right now. Basically designing the logistics and funding these larger studies to validate these algorithms.
So I'm curious, when you look across these images of the brain, even if it is a smaller sample size, how different is each person's brain? I mean, how is there a lot of variation? Is there more commonality? Like, I'm really curious as to what those images look like as far as being diverse.
That's a fantastic question. So yes, every person's brain is different. And indeed, this is a very important question because when I was saying that we're building a model about brains to learn something about them and then to know how to restore these images better, well, it's crucial to understand what family of brains are you looking at to come up with these models. And so at the moment, these models are based on a healthy population of subjects. So there is variation among them, certainly, but these variations are completely normal. And these are managed by our restoration algorithms. And so every time we reconstruct the brain, this brain is reconstructed to better fit the measurements of a specific person. Naturally, if validation is important to understand, for example, how these methods would do with patients from a different clinic or from a different population, it's even more important to understand how these methods would do when now you're looking at a patient from a slightly different population, such as one where there is some disease progression that can be reflected in the brain. There are ways of doing that, though. So, for example, in ongoing research that we have, and there's this work that's about to be published very soon in a conference coming up in April or May, if I believe. What we are doing is trying to really pin down what these models of these healthy subjects are and pinning it down in a pretty precise way, in a pretty precise analytical way, so that then whenever you have a sample, whenever you have an observation from a patient that perhaps came from a different population, we can detect that and we can understand that this patient now comes from a different distribution of patients from the ones that we've seen during training, during development of these algorithms. So your question is spot on. It's fundamental to understand these amounts of variation and how much variation is within the populations that you've seen during training and how much variation characterize perhaps something that you should be flagging as abnormal.
And is there, if you were to, just kind of extrapolating out for the technology, if you were to start taking these scans on a 20-year-old and then a 30-year-old and then, the 40-year-old, when they became these ages, would we see differences in our brains over time?
Yes, absolutely. Absolutely. Our brain evolves like any other tissue and any other organ in our body. When I was talking about biomarkers and I was describing potentially developing biomarkers to detect or associate risks with specific diseases, there are biomarkers for aging as well. This is a field where I know very little about, but some of my colleagues, for example, Jude Phillips here in the biomedical engineering department, is someone who cares very much about understanding biomarkers of aging. So indeed, a lot of these changes, some of them anatomical, some of them molecular, can be used to understand the healthy process of aging in a person.
So we talked about your recent publication, and you hinted to another publication that's coming out. Can you talk about your lab in more general terms? What is the body of work, the body of science that you are focused on?
Yes, absolutely. So my lab is a mixture of biomedical engineers and computer scientists and applied mathematicians. And we care about designing machine learning algorithms. So these algorithms that can learn automatically from data to solve specific tasks. But because some of these tasks that we care about, like in biomedical imaging, are very sensitive and and very important, we also care about aspects that have to do with how reliable these algorithms are, or how trustworthy they are, or what do they tell us about their inner workings? What do they tell us about the reasons as to why they are outputting a specific result as opposed to another? Or we also care about aspects that have to do with different performance across different demographic groups. It's fundamental to understand for these algorithms, for them to be developed and deployed at large, how they might affect different subpopulations that perhaps are minorities among the broad population. And so we sit on that interplay between applications, but also the theoretical understanding of these tools. We care very much about deploying these things in the clinic, as with this collaboration with my colleagues at the Kennedy Krieger Institute, but also we care very much about developing analysis and theoretical guarantees for these methods to try to understand when they will work, when they will not work, how much perturbation could they withstand before we stop trusting them as much. And so it's a very fun group of students. I think every time I need to pinch myself a little bit because it's really a fantastic group of people to be working with.
Yeah, that's amazing. You mentioned that you found your way to Johns Hopkins, through the steps of your formal education, and you use the term it's a perfect playground. What makes it a perfect playground there at Johns Hopkins?
So on the one hand, you have these group of amazing medical scientists who are really experts in every single one of their fields, who not only are fantastic in the science and the medical care that they provide for their patients, but they are also very inquisitive and very, very curious about solving the problems that they readily identify in their respective fields. And there's this very fluid communication at Johns Hopkins between physicians in the School of Medicine and physicists, for example, in my case, who work on MRI imaging, and engineers in the School of Engineering, where I'm primarily appointed. And so this creates an environment for discussing all kinds of problems, sort of as a breeding ground for innovation. And this should be coupled with the amazing students that we can recruit in the School of Engineering. And so you have access to these fantastic, fascinating, hard problems that are very much rooted on serious problems in biomedical data science, as in my case, but also the set of resources, including students, including infrastructure, computing infrastructure, that really allows us to play at this intersection between coming up with engineering solutions, being very mindful of what are the aspects of these solutions that are critical for for these solutions to really be deployed in practice. And because we are sort of doing what we like and enjoying working on these problems, I think that the best way of describing all of that very long description is by just saying that it is a playground.
It sounds amazing. So, how important? Because you've used the term collaboration, your collaborations. How important is it to collaborate with commercial companies with the technology and the advancements, or are the advancements coming out of places like your lab and Johns Hopkins and commercial is just kind of getting it to market, if you would. But how important is that? Do you work with commercial companies?
Thank you for bringing that up. Yes, I think companies and private entities and institutions have a very important role to play in the modern ecosystem of technology development. I think that the role of these companies is crucial, but not necessarily throughout the span of the entire development process, but rather typically after a certain point. So, for example, In the case of this specific project that I was describing before, this was really born out of conversations with radiologists and conversations with physicists, experts in MRI, who have long basically believed that doing this kind of imaging would be possible with reduced number of orientations. And then we brought our expertise and techniques from data science and from learning about data and brought this to bear. For that, really, the role of private companies has not been really that important. We developed this with the idea of providing a solution to a pressing medical problem. Now, of course, after you have a prototype for a solution like this, which we now have, then certainly, by all means, there are a number of companies that could be very much interested in developing this technology further to sponsor these validation studies that I was describing before. We are beginning to consider this. I have worked with private companies in some other different projects completely unrelated to this specific imaging problem. But yes, this is very much of interest to me and to my collaborators.
Yeah, technology surrounds us all just in our day-to-day life. And sometimes we lose track, I think, or not appreciate how long it takes to do the research, to understand the science, to develop the technology, to have it accessible to all of us. It seems like it's a very long process. Do you have, can you shed some insight into that?
Unfortunately, no. I agree with you. But at the same time, I would also say that these processes might be becoming shorter than what we are used to. Oftentimes, when this technology is really solving a very important medical need, then the private world is really ready to invest aggressively and to get these tools to market. One clear example of this was the development of the COVID vaccine that went from an idea and a research endeavor to market in record time. Of course, I don't work in the immuno world. I don't really work with vaccines, but this is just to say that the time to market, it is long, and it is long because there are very precise and thorough validation processes that have to be taken care of. But I'm hopeful that those times will continue to shrink as we move forward with technology as well.
Johns Hopkins is located very close to Washington, D.C., a lot of policymakers, regulatory. Where does policy and regulations come into this? The technology, the scanning, the understanding of that technology with the human, that connection, where does that play? And how does that help or sometimes get in the way of what you do?
That's a terrific question. It plays a fundamental role. In fact, one cannot really talk about the speed or the time that it takes for a technology to be developed and brought to market without talking about regulation, particularly in the healthcare space. So being close to DC is really a very important benefit that we have by working at Johns Hopkins. It's relatively easy for everybody here at Hopkins to just take the train down and go to downtown DC. Johns Hopkins has campuses in the very much at the heart of DC, a few hundred feet from the hill. And so there is a relatively robust and even growing relationship between everybody at Johns Hopkins and the regulation community, the policy community in Washington, D.C. I particularly care a lot about these aspects of regulation. In fact, there are many very important open problems whenever one talks about deploying tools that are based on learning automatically from data into the wild. This is still very much a ongoing conversation. It's very dynamic. It's very often a very heated discussion as to how these algorithms should be regulated, should be validated, how should the live process of an algorithm that was learned automatically from data and perhaps needs to be updated, what are those validation protocols going to look like? Or if these algorithms are perhaps suggesting, for example, different diagnoses options for healthcare providers. What kind of level of trustworthiness should they have, or what does that even look like? What kind of questions should the clinician be able to ask about the algorithm, and how should these answers be presented? So a lot of these questions that are very pragmatic, fundamental questions lead to very interesting academic questions that we care very much in my group. So we care about how these questions of interpretability of these very complicated algorithms is posed. We care about how different notions of demographic fairness could be measured and could be estimated, and potentially could be estimated when we have incomplete information about the world. So I think that to answer all of these questions, communication with domain experts, in this case, people in physicians and clinical experts, together with policymakers, together with regulatory agencies, together with people in the academic community, and in particular in statistics and machine learning and biomedical data science, a conversation with all of those actors is really fundamental to make progress along these directions. And these questions are very much open. These discussions are ongoing, and being close to DC is really a benefit that allows us to make progress on this domain.
Yeah, and where does privacy of one's data around their individual brain, because if the algorithms are learning from the set of scans from the brain, where does privacy and HIPAA and all that come into play around an individual's rights or their image?
That's a great question. So you're correct in mentioning HIPAA. So HIPAA is a set of regulations that precisely specify what kind of privacy concerns, or what kind of privacy conditions and measures should be put in place whenever we're dealing with healthcare data from patients. Now, there are settings where one can, and what basically this is not limited to just imaging data, but whenever algorithms are used, or rather are developed using biomedical data from other patients, there are ways of learning things from this data while making sure that we cannot infer too much about the data that these algorithms were trained on. So some of these, again, this is a very nice example of a problem that arises at the intersection between, for example, clinical applications and computer science and statistics. And to some extent, it's been well understood what kind of algorithms we can use to protect the privacy of, for example, people participating in a study. In fact, this is not true just about healthcare, but it is also true about data that is collected, for example, through cell phones and any other sort of end user points that private companies have to collect data. And so this question of privacy, even beyond healthcare, has been around now for 10 or 15 years. And I would love the path that the privacy world, or the privacy question, has taken to now be reflected on some of these other more modern questions that I was describing a moment ago, for example, that have to do with interpretability.
Mm-hmm. So you could tell that you kind of lit up when you talked about the young people that are working in your lab, and they're starting their careers, and they're building that foundation to kind of follow your footsteps and grow. What for those young listeners that are in research or thinking about going into research, what are your thoughts or your just your advice for them?
That's a very interesting question. Also, I would say I really don't expect, and I really don't want my students to follow in my footsteps. I want them to create their own footsteps. I think that it's hard to imagine. I think for people who have never really been exposed to an academic setting, what it really means to do research and what it entails and what it might look like. I'll tell you, I certainly had no idea that such a job existed when I was growing up. I grew up in Argentina in a relatively small city. where the concept of research was really very foreign. It was really not that well understood. And increasingly with time, I basically realized that, hey, I could actually spend the rest of my life and make a career out of this business of just asking questions and finding solutions and having fun in the process. So research is really about that, learning how to ask the right questions about a problem, and if these questions are good, then oftentimes they lead to solutions, or if they don't lead to solutions, at the very least they lead to us understanding something new that hopefully down the line will then teach us something, and will tell us how to come up with these solutions. I think that for anyone who might be interested in research, I think that the single best advice that I can voice is to just keep being curious. Really what we do is driven by being curious. And this reflects in us talking about problems with other colleagues who have a very different background than us, because we don't understand their domain and they do. And so they can teach us something. It has to do with really pursuing threads and questions where we feel that there must be something that can be done and we can't quite pinpoint exactly what. But if you think about it long enough, then you end up understanding either what that thing is or what is missing that might help you put that pinpoint there. And so I think that being curious is really a big, big, big part of this business of doing research and having fun in the process.
So, what are you curious about for the future? And what I'm trying to get to is give us some of your thoughts or insight into how the technology that you're working with or around, how's that going to change in the next 10 or 15 years? What do you see and what do you hope for, especially when it comes to solving these hard problems between, technology and humans?
So if there's something that we've learned out of the last sort of 10, 15, 20 years, is that it is extremely hard to make predictions about where technology is going to go. I think very few people expected the shift that happened in the computer vision community in 2012. I think very few people envisioned the shift that's happened with the appearance of large language models and the kind of solutions that they allow for over the last couple of years. So it's extremely it's very, very hard to make predictions. So with that disclaimer, I would love to see a setting where all the data that we are gathering about ourselves, and there's lots of this data. There is lots of data through wearables. There's lots of data through interactions with computers of all kinds. There is lots of data that is collected on regular medical exams and checkups. And then, of course, there is data that is collected in more sophisticated through more sophisticated techniques and protocols and instruments. But I would love to see a setting where all of these data can be integrated into understanding every person better, so that we can make really personalized recommendations and really sort of understanding the risks that each of us has in terms of developing certain diseases, so that we can inform not only treatments, but also inform prevention of some of these diseases, particularly in today's world, where we have diseases that sort of have correlated with the advancement of society and quality of life in general. Prevention becomes increasingly important. And so I think it's fundamental moving forward, develop tools that allow us to understand what is best for every single person. Of course, that will involve things that we don't know how to do yet. It touches on aspects, for example, of privacy that you were mentioning before, but it touches on many other considerations and problems that sit at the intersection of technology and societal constraints, and constraints that we, the society, put in place to make sure that our algorithms behave the way that we expect them to behave, in ethical ways, that they behave in ways that are aligned in some sense with what we believe as people should be done. So I would love to see all that data that we're generating being used to those ends moving forward.
So, what do you wish people knew that really may not be familiar with research or just anything that we've really kind of talked about here today? What would you hope or wish that they knew about what you do day to day?
That's a tough question. I think what people should know is that people like me and my collaborators who design technology to try to solve problems, be it in a medical setting or not, really are trying to contribute to society by providing a solution that doesn't exist and that will hopefully alleviate an unmet need that we as a group of people living in this world have. There is a lot of work, a lot of prior work that we use to come up with these solutions. It's not that we sit in our room just thinking and staring at a piece of paper until all of a sudden a light bulb lights up and then we have a solution to our problem, but rather this, coming up with these things demand a lot of collaboration, a lot of discussions, a lot of meetings, a lot of back and forth, a lot of mistakes. Sometimes we pursue directions that don't work, and we have to go back, and that is why it's called research, right? Otherwise, it would be just search. And this process of doing research and pushing innovation can get a little bit messy in that sense. It's far from a passive purely, thinking mode of finding solutions. And instead is a very active, dynamic series of activities in this thriving place with a bunch of people with very rich, different expertise, very different backgrounds who are also hopefully contributing with different original ways of thinking about problems. One thing that we haven't touched on really is that in these environments, there is truly people from all around the world with very different cultural backgrounds, very different mother tongues, very different customs, and all of that is also crucial to come up with creative solutions to problems. Sometimes there are things that will go unnoticed for certain people because we're used to looking at a specific setting, a specific problem in the same way. And it isn't until someone comes from a completely different part, either of the academic environment or from a different part of the world, and points to something that we had missed. And that then allows us to move forward. So I think that is the image that I would try to convey about what is it that we really do in our day-to-day work.
That's fantastic. And I do appreciate you bringing up the nature of collaboration across different cultures, different countries, different kind of people, different kind of thoughts, because, that's going to just add to our knowledge about us as individuals and how our bodies work and how our brains work. And maybe someday we'll get to that vision that you painted about all this data being very customized for us as individuals, so we can proactively stay healthy and keep these bodies that we live in and our minds healthy. So as we wrap up here, how can people get a hold of you? Like, tell us how they can contact you, because collaborations, you've talked about the collaborations being important. I'm sure there's people out there that are listening that would love to collaborate or have pieces of information that might be useful to you.
Absolutely, yes. So I'd be very happy to talk with anyone who's interested about exploring possible routes for collaboration. They can find all of my contact information in my website, which they can find by directly going to Johns Hopkins Biomedical Engineering Department website. So if you just look for biomedical engineering, Hopkins, and you go to the faculty site, you will find my contact info and other details as well.
That's wonderful. So any last thoughts that you want to add?
I would just like to thank you, Deb, again for the opportunity to talk and to reflect on some of these questions that have to do with how we do the work we do and why we do it. Also, the Toffler Foundation has been very generous in supporting part of our research and has really enabled us to contributing to solving that one problem and then moving on. And as I mentioned, there are many new ideas that we are now working on that have sprung out because of that initial project that the Toffler Foundation was involved with. So I'm very thankful to it, and I've really enjoyed the conversation today.
Yeah, me too. Me too. You're doing some fascinating work and very useful work and effort for all of us in understanding. So I appreciate your effort. I appreciate your dedication, and thank you so much today for your time. So thank you.
You're very welcome, and thank you too.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.